we're probably going to get right into the session. So welcome everyone to our Great Greens Chromatography webinar. We're going to be doing a live stream experiment today. If you folks do not have the ingredients with you or do not have all of the ingredients with you right now, do not worry about that. You do not have to do the experiment with me right now. And you attempt to follow along, you're probably going to be a bit rushed. So it's probably best if you wait until after the session, until the recording is sent out, to try it yourself. Because then you have a lot more time and you can really get into the details of the experiment without worrying about a time limit. So I just want to establish a couple of rules for our live stream today. There is our little chat box over on the side here that everyone is talking in right now. I'm going to be asking you guys some questions, so feel free to throw your answers in there. Don't worry about the hand up, hand down option. We don't have to play with that today just because we're going to see everyone's responses in the chat box. Please keep the chat nice and appropriate. So we should be talking about the experiment at hand today or talking about questions that you have for me. We want to treat this like this is an actual classroom and I'm the teacher, so we're going to keep it nice and appropriate. And it's also being recorded, so you do not want to say anything inappropriate because I will be able to see that afterwards. Alongside me today running this experiment, I also have another moderator who is an instructor in the STEM camp, which is my coworker Tanner. So he's going to hop on and say hello to everyone really quickly.
Hi, everyone. So I'll be a moderator. So if you have any issues or anything, just type it in the chat. Hi, Josh. And I'll let you guys know, and I'll try to help you out and work through it. Thanks.
Cool. Thank you, Tanner. I think that is all the rules I have for you guys to follow. Just keep the chat appropriate, and if you have any questions, be sure to shout them out, and I will try and answer them to the best of my ability. So, like I said, we're going to be doing an experiment. It's going to be an experiment where we conduct chromatography today. Does anyone know what chromatography means? Throw it in the chat if you have any ideas. No one knows so far. That is okay. A pretty advanced topic. So wait a few more seconds to see if anyone has any guesses on what it could be. So Sadie, you say your sister knows. Mercer, guess that it's the study of plants. It's not the study of plants. The study of plants is known as botany, but what we're doing today does involve plants. See, a couple people say their family members know. That is pretty cool. Not a lot of people know about this area of science or this experiment in science. So what chromatography means is you're just separating a mixture into its different parts. And we use two parts, which is the mobile phase and the stationary phase. Does anyone know what mobile and stationary means? Or any guesses? If you have any guesses, feel free to just throw them right in the chat. No guesses? No one knows what it means? So what mobile means, it just means it moves. Stationary just means that it stays still. So what we're going to have today is a liquid, which is going to be our mobile phase, and a piece of filter paper, which is going to be our stationary phase, meaning that it's not moving. So the mixture we're going to kind of be working with today has chlorophyll in it. Does anyone know what chlorophyll is or what chlorophyll does? If you know, type it in the chat. All right, I see a few folks are on the right track here, saying that it's green, it's stuff that gives the plants food, it makes the plants green, it makes food from the sun. Those are all Great guesses, and you're all on the right track. I see Mercer says it traps light energy from the sun, it makes leaves green. Those are all perfect answers. So what my definition is chlorophyll is the green pigment in plants. So like you folks said, it does make the plants green, and it captures the sun energy or the sunlight to help the plants make their food. So good job on folks for getting that correctly. So the plant that I'm going to be using today in our experiment is spinach. So spinach is obviously green because it contains chlorophyll, but there are some other pigment molecules in plants and even spinach that we might see today. So we have like chlorophyll, which is green. We also have anthocyanin, which is on the reddish stem. Then we have carotenoids, which are orangey in color. And then we also have xanthophyll, which is yellow. So even though our plants appear green or our vegetables appear green, there's lots of other colors and pigments that exist within them. So we are going to be extracting chlorophyll today, and chlorophyll can actually be broken down a little further into chlorophyll A and B. There's two different types of it, and we'll see how that comes into effect a little later. So chromatography can be used in a variety of areas of science. Can anyone guess what fields of science or what topics you might learn chromatography in, or you might use chromatography in? Or maybe even some jobs? If you have a guess, just throw it in the chat. Biology, absolutely. So biology, you would probably use chromatography to understand a biological or organic mixture. Any other fields you folks can think of? There's one main one. Chemistry, yes, good job, Mercer. So it is used in chemistry a lot to find out a lot of different things, because in chemistry, it's all about mixtures and it's all about solutions. So finding out what is in those solutions and mixtures is really important, and chromatography is one way that they do that. So Sadie was wondering if farmers use chromatography. It's really hard to say if farmers use it. I'm sure on perhaps more of an industrial scale, they probably have some scientists that work in a lab somewhere in their area that probably help with the farm and identifying things like maybe fertilizers that they use, or how their plants react to different substances. So definitely a possibility. I see someone said botany. I'm not too sure if botanists use chromatography. I'm sure they're definitely intersectional, so you can probably get botanists that do chromatography and botanists that don't do chromatography, depending on their background. So maybe if they have more of a chemistry or biology background, they're going to be doing some chromatography in the lab, perhaps. Another area that you folks might not have thought of is forensic scientists, forensic pathologists, any sort of forensic analyst. They use chromatography a lot. Forensic science is one of my favorite areas of science, and it's what I hope to go into in the future. So maybe in the future, I will be doing some chromatography in the forensic fields. Another activity that I've done in the past, not related quite to jobs, but something that I've actually done at Supernova. Maybe if you were in the summer camp for science this summer and had me as an instructor, you'll remember that we separated black ink using chromatography. So if you want to do this experiment at home, what you can do is you can take a black marker, draw a little dot or a big dot, however big you want it, on a piece of paper towel and dip that in some water. And as the water passes through the paper towel, it's going to split black ink into a bunch of different colors. So it's actually not just black. You're going to see blues and purples, greens, yellows and oranges. All those colors are going to split out of it. So the reason why we see these layers or these bands of color form is due to different polarities. Does anyone have any idea what polarity is? Any guesses on what polarity is? You can throw it in the chat if you have a guess. I guess magnets, that is a really good guess. So magnets do have a north and a south pole, which is kind of related to what polarity is in this context. So what polarity means is that there is going to be a positive end and a negative end to a molecule. So it does create a difference in charge. So similar to a magnet, the different pigments and different color molecules that we're going to be dealing with today have different polarities. So when we pass a mobile phase, like a liquid through it, it's going to separate depending on its polarity. See, someone said batteries. That is definitely another example of polarity because they have a positive and negative end. So some molecules are polar and some are nonpolar, meaning some of them do have a difference in charge and some of them don't really have a difference in charge at all. Getting into the ingredients I'm going to be using today, the solvent, liquid solvent, or the mobile phase that I'm going to be using is one of my favorite ingredients to use in science because I use it all the time, isopropyl alcohol. So if you were part of the live stream last week or the STEM club that's been going on, you'll know that we have been using isopropyl alcohol for a few things. And it is polar, which is what we want to use today because it's going to help pull all of those pigments apart. So you need isopropyl alcohol. Another thing you're going to need is a short glass and a tall glass, or a short container or a tall container, whatever you have around you that works. You just need something short and something tall to put your ingredients in. You also need some leaves. So I mentioned spinach today. What I did was I went out to the grocery store and I picked up this half and half salad mixture. So it's half spinach and half baby greens. And the fun thing about it is that not all of the leaves in there are green. Some of them are purpley, red, and pink, which means they probably contain anthocyanin in them as well as chlorophyll. So for today's experiment, I'm just going to be using spinach. So I took a bunch of spinach out of that bag and separated it. You are also going to need a piece of coffee filter paper. So this is just one piece. You can use as many as you want depending on how many solutions you create. You also need a deep dish. So I put in the materials list, you probably have a baking dish around, you can use that. But for my experiment on camera right now, I'm just using a bowl, a deep bowl, because that's what we're going to set our solution into. You also need a pair of scissors. So nothing fancy, just a normal pair of scissors, because that's what we're going to use to cut our coffee filter paper. And you also need some hot water. So if you're playing with hot water or using hot water in an experiment, it's probably best to have an adult help you with that. So note that if you're going to be using hot water, get an adult to help you with that step. So I'm going to get into the experiment and explain the steps as I go along. So what do you folks think step one is? Any guesses what I'm going to do first? Well, I'll tell you. I am going to rip up a bunch of spinach leaves. So this is pretty fun. Not something that you would typically do with spinach because you usually eat them. So what I'm going to do is I'm going to start breaking up all of these leaves into small confetti-sized pieces. And I'm just going to put them into my short glass. So I'm breaking up all of these leaves into tiny, tiny, tiny little pieces and putting them into the short glass. And you can include the stems of the spinach leaf if you want. You don't have to. I am just for simplicity. I'm continuing to break these up into tiny little pieces and putting them in my glass. The reason why we are breaking up the leaves is because when we add our solvent in a later step, it's going to increase the surface area that the solvent is acting on and breaking down those leaves. Because remember, what we're trying to do is we're trying to get the chlorophyll out of the leaves. So in order to do that, we need to break them down and break down those plant membranes and get all of the pigment out. And it's a lot easier if our leaves are broken up into tiny little pieces. So I'm just gonna break up a couple more leaves here tiny, 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 tiny pieces and put them in the short glass that I have. So I have a good amount of leaves in here, tiny little layer on the bottom. And now what I'm going to do is I'm going to add one of my favorite ingredients, which is isopropyl alcohol. You do not need a lot of alcohol for this experiment. If you're going out to buy materials after this video, just get the smallest bottle that you can find because there's really no need in getting a huge bottle. So what you want to do is add just enough isopropyl to cover your leaves. You don't want to fill your glass with it by any means. You just want to cover it right at the bottom. So what I'm going to do is I'm going to add that in now. Pour that in. So you can see I don't have a lot of isopropyl in there. It's just enough to cover the leaves at the bottom. So what that does, it helps us create a really concentrated solution, which is what we want. Another step you can do right here, if you don't think that breaking up your leaves is enough, is you can start to mush your leaves up using, in my case, I'm using a spoon. So I'm just taking a spoon and I'm mushing up my leaves at the bottom of the cup. Like before, mushing it up and breaking them apart helps break them down a lot quicker and helps us get all of that chlorophyll out, which is what we want. Put that aside for now. So I mentioned hot water, and that is what our next step is you actually need to add hot water to your bowl or your dish that you're using. So I've already done this, so I just have some hot water in this bowl here. What you're going to do is you're going to take your solution of isopropyl alcohol in leaves and just place it in the bowl or dish of hot water. Can anyone guess why we place the solution in hot water? It relates to what we've done by breaking up the leaves. So it happens faster, so they react with something. Those are definitely on the right track. So what happens is the heat actually helps bring out the chlorophyll into the solvent a lot faster. So solvent is doing its job as a solvent in breaking down the spinach and breaking down the chlorophyll, but heat helps speed that up and helps it happen a lot faster, a lot better. So think about when you maybe see your parents steep some tea or make a mug of tea. You add the tea bag in with some hot water and you let that steep for a little bit, and all of the tea comes out of the leaves into the water. It doesn't work so well in cold water and it takes a lot longer, and that's why folks use hot water in their tea, and that's why we're using hot water with our spinach today, because we're allowing them to steep and allow all that chlorophyll to come out. So you do have to leave this for quite a long time. The longer you leave this, the better. I left mine for 24 hours. You can leave it for a few hours. You can leave it overnight, whatever is easiest for you. But I'm not stopping the experiment right now because I did this last night to show you folks what it looks like today. So what do you think the solution looks like if you leave it in hot water overnight? Any guesses? Jen thinks it's going to be completely green. That is 100% correct, and the answer I'm looking for. So what happens is the isopropyl alcohol has all of this chlorophyll in it now, so it's going to be a really, really, really green color. People think the leaves are going to turn a different color. That can absolutely happen. So when you add the leaves to the isopropyl alcohol, you'll notice that they're going to get darker because they're starting to be broken down, and the spinach isn't too happy about being an isopropyl alcohol. Also, if you leave them in for a really, really long time, you might notice they're not as green as before because all of their pigment molecules are being steeped into the solvent. People think they're going to become squishy. Absolutely, they're going to be really squishy and mushy, especially after you've broken them up and let them sit in liquid for 24 hours. So I'll show you what that looks like. Like I said, I've already done this. I did this yesterday and left it for 24 hours. And now my solution is this super dark green liquid. So the hot water helped steep all of the pigment molecules out of it, and the solvent helped break down the plant membranes. So we get all of this chlorophyll floating around in our solution. It looks almost black. Yeah, it's so dark it looks almost black, but it's actually a very, very rich green color. If I put it close to the camera, you can see how green it is. So the next part of the experiment is actually creating the filter paper or the filter strips that we're going to use to separate the pigments. So this is where your coffee filters come in, and your scissors. What you want to do is just cut a strip out of the paper. You can cut as many strips as you want, but since I only have 1 cup of solution, I'm only going to cut 1 strip. So it can be as big or as small as you want. Just going to cut this up really quick. And put my scissors aside. And what I'm going to have now is just a rectangular strip of filter paper. So a big part of chromatography is using chromatography paper, which we put the liquid mobile phase onto and then put it through the solvent, which pushes it up. So remember, this is the stationary phase, and the mobile phase is going to run along it and separate the pigments that we want to see. So the next step can also be a little time consuming. What you want to do is, I just used the spoon that I mushed up the spinach leaves with earlier, and you want to drip a little bit onto the bottom end of your paper. And you're going to see it's going to absorb and it's going to suck up all of the green solution. That's good. That's exactly what we want to have, but we have to let that dry. And after you let it dry, you want to repeat that step. So you want to take your spoon or maybe even a toothpick or even dipping it in the solution once it's dry and getting more of that green pigment onto the end. You want to do this 2 or 3 times. So you want to get the pigment on there, you want to let it dry, and then repeat the process 2 or 3 times, because what we're doing is we're creating a super concentrated green area of the filter paper. And then once you do that, you're going to let it dry for the final time. And this is where your tall glass comes into the experiment. So I'm going to grab a tall glass or a tall container, whatever you're using, and a little bit of our favorite ingredient, isopropyl alcohol. And you're going to pour just a tiny, tiny little bit into the bottom of your tall glass. So I'm just going to pour literally 1 splash in. So we just have a tiny layer in the bottom. So remember, our liquid ingredients here today are our mobile phase. So they are the things that move. I'm just going to get my ingredients out of the way. What you want to do now is you want to take your filter paper that has your green end. So remember, you've dyed it a couple times, allowed it to dry. Now you have a super green end. And you want to dip that end into your tall glass and let the end of it, that's green, touch the liquid. So touch the isopropyl alcohol. What's going to happen then, the isopropyl alcohol is going to be absorbed into the paper, and it's going to start rushing up the paper. And as it rushes up the paper, it's going to pass through that pigment that we had, and it's going to start separating the pigment into bands, which is what we want to see. You're not going to see this until after it dries. So it might be helpful to fasten the top of your paper around your glass. You can by just like folding it over so it stays up, or even just taping it in place. And after that dries, that is when we see the end result of our experiment, which is what we want to see. And I can show you folks right now because I did this last night. So the very bottom of my filter paper is green, but you can kind of see lines forming right at the bottom. And that's because chlorophyll A and chlorophyll B split apart at this layer. and created a separate light green and dark green. So we do have our chlorophyll green molecules, but remember earlier we talked about chlorophyll A and chlorophyll B being different. They also have different polarities. Remember we talked about polarity being the partial charge or the opposite charged ends of a molecule. So what happens is due to one reacting a little bit differently with our mobile phase, it traveled a little bit further than the other chlorophyll molecule. This isn't the only thing I observed in this paper or in this experiment though. Up here, you can't really see it. I'm going to get it nice and close to the camera. There's a little yellow band right here. It's super hard to see on camera because it's super faint, but you'll be able to see this if you do it yourself. We act on that there's a separate pigment molecule on the paper, which is the carotenoid. So it's this yellowy orange color. So on top of the chlorophyll, we also had carotenoid pigment molecules in our spinach. And by allowing the mobile phase to pass through it, we saw it was drawing all the way up to the middle of my paper, which is far away from where the chlorophyll was. So the results of this experiment show that I had 3 different pigment molecules in my spinach. I had chlorophyll A, I had chlorophyll B, and I had carotenoids. And that's the basis of what chromatography is taking some solution, some mixture, and we don't know what's in it. We have an idea, and we pass our mobile liquid phase through it, and it separates into the different bands because of the different polarities. So if you folks do this, I suggest using a bunch of different colored greens, a bunch of different colored leaves, and seeing what different pigment molecules you can see. Remember, this does take a little bit of patience because you do have to let the paper dry from time to time. And let all those pigments dry on the paper in order for it to work. So you can watch this video back over to the steps and make sure you're getting everything right. I will also send out a little written explanation for you folks, just so you have a good idea on what to do. But that is kind of the end of the experiment. It is a longer one, but I did shorten it a lot for this experiment just because I did it over time already and had it for you folks. But if you have any questions for me, I will gladly take them now and answer them for you. So if you have any questions for me, just type them in the chat and I will be happy to answer them.